Scott Brown's Otology, Chapter 47, Anatomy of the Cochlear and Vestibular System relating ultrastructure to function. General characteristics of hair cells. General characteristics of hair cells. The hair bundle and cuticular plate. The hair bundle is composed of two rows of stereocilia that increase in height in one particular direction across the apical surface of the hair cell, and a single kinocilium located behind the row of longer stereocilia, but which is absent from the hair bundles of mature hair cells in the cochlea. Stereocilia are really giant microvilli, plasma membrane bound projections from the apical surface of the hair cell that enclose a packed array of filaments of the cytoskeleton protein actin. The kinocilium is composed of microtubules similar in form to motile cilia but are not motile. In cochlear hair cells, the kinocilium is present only during development, reducing down as the cochlea matures to remain only as the basal body in the apical cytoplasm at one side of the stereociliary bundle. The position of the kinocilium or the basal body and the longest row of stereocilia define the polarity of the asymmetric hair bundle. Deflection of the stereocilia towards the longest ones opens MET channels, potassium enters, and the hair cell becomes depolarized. Deflection in the opposite direction closes the transducer channels, and the hair cells become hyperpolarized. The transducer channels are located at the tips of the stereocilia of the shorter rows but not in the longest stereocilia. The parallel actin filaments in the stereocilia are closely packed and are cross linked by a number of different proteins such as espin, fibrin, facin, and plastin 1, the last being the second most abundant protein in stereocilia after actin. The high density of actin filaments and the extensive cross linking between them imposes rigidity on the shaft of the stereocilium, which tapers significantly at its proximal end, where it is embedded into the cuticular plate. A rigid platform formed of a meshwork of actin filaments and other proteins in the apical cytoplasm of the hair cell. As a result, when pushed at its tip, the stereocilium pivots at the taper like a stiff rod. The fundamental nature of this stereociliary rigidity for all hair cells is evidenced by the hearing impairment and vestibular disorders that result from mutations in the genes that encode for such actin cross-linker proteins. Mice with such mutations are often reorganized by erratic and or encircling behaviors. For example, the jerker mouse strain, which has a mutation in the gene for the cross-linking protein espin, and in which stereocilia are thinner than normal. Loss of plastin 1 results in progressive hearing loss and balance dysfunction and progressive thinning of stereocilia. Actin filaments descend from the stereocilium into the cuticular plate as a rootlet which is cross-linked into the actin meshwork. The rootlet is formed of densely packed actin filaments. The actin bundling protein TRIOPB plays a key role in the formation and maintenance of the rootlet. In addition to actin, the cuticular plate contains spectrin, another actin cross-linking protein that has elastic deformation resisting properties, and tropomyosin, a protein that binds around actin and stiffens it. Around its lateral margin, the cuticular plate is linked to the lateral plasma membrane at the level of the intercellular junction, which On the supporting cell side, actin and other cytoskeleton proteins are also associated. This may provide a means of support for the cuticular plate so that the stereocilia themselves are supported on a rigid platform, enhancing their ability to respond to small displacement forces. Various members of the myosin family of motor proteins types 1C, 6, 7A, and 15, all unconventional non muscle isotypes, are also localized in the cuticular plate region and the stereocilia. In addition, immunolabeling for myosins 6 and 7A shows these two proteins to be highly expressed throughout the hair cell cytoplasm, providing a distinguishing marker label for hair cells from their earliest differentiation during development and into adulthood. Mice with mutations in the genes for myosin 6, 7A, or 15 all show deafness and balance disorders and abnormalities in their stereociliary bundles. In the mouse mutant, where myosin 6 is defective, Snell's Waltzer stereocilia are fused and greatly lengthened. Interestingly, a similar anomaly is seen in the human organ of Corti and vestibular system from elderly people and may be related to age related hearing loss and balance dysfunction. It has been suggested that myosin 6 may be involved in holding the apical plasma membrane of the hair cell onto the cuticular plate in the regions between stereociliary bases so that individual stereocilia can be maintained, and when it is defective, that membrane region becomes detached. In mice where myosin 15 is mutated, Shaker 2 stereocilia are greatly reduced in height. Myosin 15 is thought to form a complex with other proteins including verlin and ESP8 that regulates the height of the stereocilia. Mutations in the myosin 7A gene are responsible for Usher syndrome type 1B. The mutant mouse strain carrying this mutation, Shaker 1, shows hair bundles in which group of stereocilia are separated from each other at the hair cell apex, and the kinocilium is misplaced, suggesting an effect on maintenance of orientation and interstereociliary stabilization. Myosin 7A may have a role in the various cross links that are present between stereocilia. The stereocilia in an individual hair bundle are connected by a variety of fibrillary extracellular crosslinks. The most critical of these is the tip link, which runs from the top of one stereocilium to the shaft of an adjacent longer one along the line of morphological polarity, and thus generating the line of function polarity. The tip link is thought to be the gating element that controls the opening of the MET channel. As the bundle moves in the excitatory direction, tension on the tip link opens the channel. When the bundle moves in the opposite direction, tension is relieved and the channel closes. The tip link is composed of two coiled filamentous proteins that are adherent end to end. Catherine 23, CDH23, and Protocatherine 15, PCDH15. Unsurprisingly, mutations in the genes that encode for either of these proteins cause both hearing impairment and vestibular disorders. Mutations in the CDH23 gene result in Usher syndrome type 1D in humans and are responsible for the Waltzer mouse strain. Mutations in the gene encoding PCDH15 are associated with Usher syndrome type 1F and the Ames Waltzer mouse. The names of the mouse strains indicate the movement abnormalities resulting from the defects in the vestibular system. At its upper end, around that point of its insertion into the membrane of the shaft of the longer stereocilium, the tip link is associated with a complex of proteins that may regulate tip link tension, thereby controlling transduction channel opening, as well as adaptation, the closure of the channel that occurs with sustained excitatory deflection of the stereocilia of particular relevance in vestibular hair cells. Proteins involved in maintaining tip link tension include myosin 7A, SANS, and harmonin, and again, defects in these are associated with hearing impairment and vestibular dysfunction. Usher type 1b in humans and the Shaker 1 mouse strain. In the case of myosin 7A, Usher type 1c from defects in harmonin and Usher type 1g from mutations in SANS. In addition, myosin 1c localizes to the region near the upper insertion point of the tip link and is thought to be involved in adaptation. In addition to the tip link, there are lateral links that connect the shaft of one stereocilium to all its neighbors. There are thought to be at least three different types of lateral links between stereocilia. Ankle links, which are present in the hair cells of mammalian vestibular organs, and the auditory and vestibular hair cells of non mammalian vertebrates, but not in those of the organ of Corti, connect stereocilia at their proximal ends. Shaft connectors are present along the mid region of the stereociliary shaft. The top connectors link stereocilia just below the level of the top tip links. The different types of lateral link were initially identified through the use or generation of antibodies that specifically label each subpopulation separately. This indicates significant differences in protein composition between the links. Mice with defects in the genes for these different stereociliary cross linking proteins also have hearing impairment and balance dysfunction. Lateral links may have a role in holding the bundle together, thereby stabilizing it and aiding the mechanical coupling of deflections of the stereocilia, such that the stereocilia in a hair bundle all move as a single unit. In agreement with this, mice with mutations in genes that encode lateral link proteins exhibit splaying of the cilia and thus loss of efficient MET, indicating the critical role for lateral crosslinks in maintaining hair bundle cohesion. The basolateral plasma membrane. The basolateral plasma membrane of a hair cell is characterized by the presence of a variety of ion channels essential for shaping the response of the cell to mechanical excitation. Outwardly rectifying potassium channels open when the hair cell is depolarized by the MET current that is generated by hair bundle deflection. The resulting outward flow of potassium through these channels repolarizes the hair cell membrane. The same depolarization also leads to opening of voltage gated calcium channels and thus an influx of calcium that triggers the fusion of intracellular vesicles with the plasma membrane and release of neurotransmitter at the synapses onto the primary efferent nerve endings. In this way, stereociliary deflection opens MET channels, causing depolarization in the hair cells, releasing of the neurotransmitter onto efferent nerve fibers, and thus activation of action potentials in neurons, that is neural discharge. In addition, hair cells in some rodent species also possesses inwardly rectifying potassium channels that are opened by cell hyperpolarization to restore cell resting potentials. Ion channels with these general properties can have different physiological characteristics in terms of their voltage dependence, the speed with which they respond, fast or delayed, and the size of the current that flows through them. Although the general features of the hair cells are well served, conserved across many species. Different types of hair cells are recognized depending on their location, auditory or vestibular, innervation pattern, afferent or efferent, and species of origin. The types, different types and numbers of ion channels express are characteristic features of particular types of hair cells. In addition to the expression of different ion channels, hair cells in organ of corti show other specializations of their basolateral plasma membrane that are associated with particular unique functions that these cells perform. We'll see the diagrams. Figure 47.3, vestibular sensory epithelium, utricular macula, scanning electron microscopy of the entire intact utricular macula viewed from above after removal of otocorneal membrane. Curved line indicates approximate location of line of hair bundle polarized polarity reversal in the stellular region. Scale bar 100 μm. B is the saccular macula as above the utricular macula, scale bar 100 μm. C is the crista ampullaris, scanning electron microscopy of the upper surface of the crista saddle, scale bar 100 μm. D is a transmission electron microscopy of a thin section of a vestibular sensory epithelium, utricular macula of guinea pig. T1 type 1 hair cell, T2 type 2 hair cell, SC supporting cell. Note that nuclei of each type are at different levels in this pseudostratified epithelium. Scale bar 5 μm. E is the human utricular macula labeled for filamentous actin, F actin, with fluorescent phallodoin in red and an antibody to calretin in green, labeling hair cell. Apical surface view to show reticular lamina. Phalloidin label F actin in the unusually wide junctional complexes around each cell. Each hair cell green is separated from its neighbors by intervening supporting cells. SC scale bar 100 μm. F is a apical F is the apical surface of the utricular macula. Hair cells are easily identified by hair bundles that project out of the surface of the epithelium. Each hair cell is separated from its neighbor by intervening supporting cells. SC scale bar 5 μm. G is the scanning electron microscopy of the surface of the utricular macula with the otocornea still in situ covering the surface. Scale bar 100 μm. H is a higher magnification view of the utricular macular surface showing the perforated sheet of extracellular matrix component of the otocorneal membrane beneath crystal like otocornea. Scale bar 5 μm. I is a hair bundle arrowed showing projecting up to the otocorneal matrix. Note that the hair bundle is aligned with the perforation in extracellular matrix component of otocorneal membrane utricle. Scale bar 5 μm. J is a cupula overlying a crista ampullaris, scale bar 100 μm. A is a whole cochlear modulus, Scanning electron microscopy showing the organ of corti spiraling down from the apex for 2.5 turns, scale bar 100 μm. B is the cross section of the organ of corti, transmission EM of a thin section of the organ of corti. The tectorial membrane TM retracts away from the surface of the organ of corti during processing. The tunnel of octi (TOC) is created by the arch formed by pharyngeal processes of the inner and outer pillar cells. Scale bar 100 μm. C. Supporting cells to the outer lateral side of the organ of corti. Scale bar 10 μm. D is the surface and underside of the organ of corti. Gerbil, scanning electron microscopy of the apical surface of the outermost lateral supporting cells removed to expose the outermost third row of Datius cells DC and the outer third row of outer hair cells OHC. The body of the Datius cells surrounds the base of the OHC and its pharyngeal processes ascends at an angle to the apical surface where the head expands. The hair bundles of the inner hair cells IHC in a single row, scale bar 10 μm. E is the luminal surface of organ of corti, mouse, hair cell or supporting cell of the cell, mosaic formed when the apical surfaces or head of various supporting cells join with and the intercalate between individual hair cells. The head of the inner pillar IP and outer pillar OP are indicated. Scale bar 5 μm. F. Human organ of corti, reticular lamina at apical surface across outer hair cell region OHC1 first row outer hair cell heads of various supporting cells surrounding each hair cell are indicated scale 10 μm. G is the tectorial membrane in situ over the organ of corti. Radial filaments comprise the body of the tectorial membrane, which has a greater density at the outer tip. The upper surface of is covered by the densely packed fibers forming the convergent CN, scale bar 5 μm. For all the panels IHC or IHC inner hair cells, OHC outer hair cell, IP inner pillar cell, OP outer pillar cell, DC data cell, HC Hensel cell, CC Cordius cell, BM basement membrane, TM tectorial membrane, BC Botchius cell, D1, D2, D3 heads of the three rows of diateous cell. Figure 47.5, the hair bundle. A is a hair bundle from uticular macula. TEM shows stereotypical features. Rows of stereocilia of increasing height with a longer kinocilium arrowed behind the longest stereocilium. The stereocilia embedded into the cuticular plate CP at their bases. Scale 1 μm. B. Hair cell bundle of outer hair cells in the human cochlea. Stereocilia in rows of increasing height but no kinocilium. In humans, there are typically more stereocilia in the hair bundle than in most other mammals. Scale bar 1 μm. Preparation courtesy Professor Tony Wright. C. Apical end of inner hair cells in mature organ of corti. The kinocilium has retracted to leave the basal body arrow to the side of the bundle with the tallest stereocilia in a region of cytoplasm outside the cuticular plate CP. D. Labeling for F actin with fluorescently tagged phalloidin red showing the stereocilia, and the cuticular plate CP are composed of F actin. Labeling for a protein that is present in basal bodies shows basal bodies located behind the stereocilia in a region free of actin labeled cuticular plate arrow. Scale bar 5 mm. E is the stereocilia in cross section showing closely packed filaments of actin. Filaments in rootlet are more densely packed. Scale bar 100 nanometer. F parallelly closely packed filaments of actin in stereocilia. Lateral cross links between the shafts of adjacent stereocilia in the same row appear to interdigitate at the midpoint of the space between them. Arrow. Scale bar 100 nanometer. G. Individual stereocilia taper at their proximal end where they embed into the cuticular plate. Scale bar 500 nanometer. H. Stereociliary rootlets are cross-linked arrowheads between before they descend into the filamentous meshwork that constitutes the cuticular plate. Scale bar 250 nanometer. I. Tip links, some arrowed, from the top of a short stereocilium to the shaft of the adjacent longer one in the direction of the line of polarity in sacula macula of Newitt. Scale bar 100 nanometer. J. Higher power view of tip links between stereocilia on outer hair cells showing branching at their ends. Scale bar 50 nanometer. K. Lateral crosslinks between neighboring stereocilia in the same row and adjacent rows. Arrow indicates apparent tip link. Scale bar 100 nanometer. Figure 47.6 hair cells and supporting cells in vestibular sensory epithelia. A. Hair bundles across the line of polarity, reversal in the striolular region of uticular macula, kinocilia arrowed behind row of longest stereocilia. The polarity of the bundles changes by 180-degree, indicated by the direction of arrows in the striolular region. Scale bar 1 μm. B is the type 1 cells. The flask shaped cell body is entirely enclosed within the calyx terminal of a single efferent nerve asterisk. Scale bar 2 mm. C. Type 2 hair cells. The cylindrical cell is contacted by several efferent nerve button terminals, arrowheads. Scale bar 2 mm. Support D. Supporting cell with the cell body basally resting on the basement membrane has a thinner process through the mid region of the epithelium and a wide, dense cytoskeletal complex at the apical head region at the level of the junction with adjacent supporting cells. Scale bar 2 mm. E is the human uticular macula hair cells labeled for myosin 7A in green. Punctuate labeling for connexin 30 red reveals numerous large gap junction plaques arrow between the cell bodies of adjacent supporting cells. Nuclei labeled blue DAIPI. Scale bar 5 μm. Figure 47.7 Ultrastructure of hair cells in supporting cells of the organ of Corti. A. Inner hair cells, flask shaped cells closely surrounded by supporting cells. Terminals of efferent synapses at the base of the cell, arrowheads. Scale bar 2 mm. B. 3D reconstruction from serial sections of the inner hair cells and its innervation. Several different efferent terminals colored and indicated by asterisks contact around the basolateral surface of the inner hair cells. Purple. Scale bar 5 mm. Figure C. Efferent nerve terminals and synapses ATEM section through the postsynaptic terminal and the presynaptic ribbon in the inner hair cell side arrowed. The circular ribbon opposes the postsynaptic density membrane of the efferent nerve terminals scale bar 1 millimeter. D is the ribbon synapse higher power view showing the ribbon surrounded by neurotransmitting vesicles. Arrowhead circular ribbon opposes the postsynaptic membrane of the afferent nerve terminal AFF scale bar 1 millimeter. D is the ribbon synapse. Higher power view showing the ribbon surrounded by neurotransmitter vesicles. Arrowhead indicates postsynaptic density of the membrane of the afferent terminal scale bar 100 nanometer. E is the outer hair cell efferent nerve terminals EFF around the basal pole of the outer hair cell. The lateral surfaces of the outer hair cell bodies are surrounded by extracellular space, the space of newel. There is contact with a datus cell around the basal pole and with the head of the datus cell at the apical surface. The head of the datus cell contains a wide, deep cytoskeleton assembly at the level of the intercellular junction and the hair cell cuticular plate. Arrowhead indicates a microtubular bundle of body of dendrite cells. Below the outer hair cell, scale bar 2 millimeter. Figure F. Underside of the tectorial membrane showing indentations of insertions of the tips of the longer stereocilia of the each outer hair cell stereociliary bundle scale 1 millimeter. G the outer hair cell stereociliary bundle. The tips of the longer stereocilia are crowned with fibrillary material shows and that attaches each stereocilium into its indentation in the underside of the tectorial membrane, probably composed of stereocylin scale bar zero point 0.5 millimeter. Figure G. Figure H. H data cell consists of a main cell body region that at its upper end encloses the base of an outer hair cell OHC, a phalangeal process that ascends at an angle or to the apical surface and an expanded head region at the apical end that contacts an outer hair cell two cells away from the row from that which the cell body encloses scale bar 5 millimeter. Figure I horizontal oblique section through the rentier cell's body where it encloses the base of the outer hair cell OHC in a cup-like fashion open on the side towards the modulus. Efferent nerve terminals EFF contact the basal body pole of the outer hair cell at the opening scale bar 2 millimeter. Figure J. Datius cell head. Wide, deep cytoskeletal assemblies arrow attached at the intercellular junction with outer hair cells, microtubular inner bundle cell head, wide, deep cytoskeletal assemblies arrow attached at intercellular junctions with outer hair cells, microtubule bundle arrowheads run across the head parallel to the apical surface and down phalangeal process scale bar 2 millimeter. K Figure K. Diateous cell phalangeal process in horizontal cross section. The process encloses bundles of microtubules scale bar 200 nanometer. I Figure I Outer pillar cell OP, the pharyngeal process is filled with parallel microtubules. The expanded apical head region forms a buttress behind the widening head of the inner pillar cell. The junction between the outer and inner pillar cell is supported by a dense, wide cytoskeletal assembly in the outer pillar cell arrow, into which the microtubule bundles terminate. Scale bar 2 mm. Figure M. Outer pillar cell phalangeal process in horizontal section. The process is filled with many closely packed microtubules in seemingly regular arrays. Scale bar 1 mm. Figure N. Inner pillar cell base. The cell body rests on a thin bony lip arrow extending out from the modulus. Microtubules bundles as descend from the phalangeal process to the very base of the cell and are associated with the dense triangular shaped cytoskeletal assembly arrowhead that anchors the cell to the underlying bone. Scale bar 2 mm. Figure 47.7. Ultrastructure of hair cells and supporting cells in the organ of Corti. Figure O. Henson's HC and Claudius CC cells. Henson cell covers the outermost extracellular space of the organ of Corti and does not reach the basilar membrane. Claudius cells are unusually devoid of organelles. They cover the Boschiet cells. Scale bar 10 mm. The Botched cells sit on the basilar membrane BM. They have a dense cytoplasm and numerous interdigital digitating infolding. Scale bar 5 mm. Q. The organ of Corti contains many gap junctions complexes. Immunohistochemical labeling for connexin 26 green reveals gap junction plaques. F actin is labeled using fluorescent phalloidin red and cell nuclei using DAPI blue. There are numerous large gap junctions between adjacent supporting cells across the entire organ of Corti. Scale bar 10 mm. Thank you.